Okay, welcome everyone. I'm the stand-in for Dr. Scott Hamilton today. And this afternoon, I have the real pleasure as the current CCFRP postdoc, or one of them, of introducing someone who really helped build the world a lot of us in this room work in. Dr. Rick Starr is research faculty here at Moss Landing Marine Labs and a former director of the California Sea Grant Extension Program. His career has focused on the biology and ecology of fish species. This includes everything from age and growth and reproduction to habitat relationships and movement ecology, always with an eye towards how that science can directly inform fisheries management. Rick was a pioneer of collaborative fisheries research long before it was a buzzword. In 2006, he co-founded the California Collaborative Fisheries Research Program, CCFRP, my current employer, with Dr. Dean Wendt to bring anglers, managers, and scientists together to monitor California's emerging network of marine protected areas using standardized hook and line surveys. Since then, CCFRP has grown into a statewide program that has conducted over 800 sampling trips, caught and released more than 230,000 fish from 98 species, and tagged nearly 74,000 fishes to understand how MPAs are working for both ecosystems and fisheries. Rick's work spans from Alaska to Costa Rica, Belize, and Spain, and has included everything from scuba and ROV surveys to submersible dives and long-term tagging studies of rockfishes, sharks, and other key species. Please join me in welcoming today's speaker, Dr. Rick Starr.
Well, thank you. And I want to say, to start with, I'm not going to talk much about CCFRP. Nate could probably talk way more about it than I can at the moment, so you'll have to wait for his seminar. And I have designed this talk to give you some ideas for the future. I remember when I was in grad school, and I thought, gosh, everything's already known. You know, there's no way I can contribute to science when I get out of grad school. So maybe I should go be a plumber or something. At least I could make some money. But I want to tell you that that's not true. And it's not true because as a lot of wise people have said in the past, the more you know, the more you don't know. And that's really the case here. Is I started out my career thinking there's no way I was going to be able to contribute much information. And now I still feel that way because there are still so many unknowns right now. So I'm going to kind of take you through what I've learned over the years and how my group has approached some of the problems that we see, and hopefully that will give you some ideas on how to approach your own careers as you go forward. So I'm going to start by saying most of what goes through my head is related to landscape ecology, because those biologists are way ahead of marine ecologists because they started 100 years earlier than we did. And they've come up with a number of different components of landscape ecology, and that includes knowledge of habitat types, patches, mosaics of habitats, the effect of habitat perturbations on animal populations, how those habitats are connected through movements, and the actual variation in movements of different animals at different life stages. And again, since they've been doing it for longer, the effects of time and change on habitats and movements. And they've come to an agreement that the watershed is an appropriate scale for managing resources, because that's a kind of a self-contained unit. So what I've tried to do is look for those kinds of analogies in the ocean to see if we could come up with some approach to understanding seascape ecology. Well, we do know a lot about fish in the ocean in California. Larry Allen and Dan Pondell and Michael Horn wrote a great book on the ecology of marine fishes in California, and have a lot of information about where fish are found. But that all comes from fishing. And so it's basically equivalent to flying a helicopter across a forest and putting a trap down and seeing what you get. You know, you might get a bird, you might get a rodent. And so that book is great. Some students, Jason Cope and Lisa Kerr, and I wrote a book on trends in fisheries in the Monterey Bay a long time ago. So we know more about fish in Monterey Bay from that. And of course, Milton Love and Mary Yaklovich wrote the Bible on rockfishes. So we know a lot, but it most again comes from fishing. And so because of that, we can tell you if the bottom is hard or if it's soft, and where those fish were caught. And so, again, 20 or 30 years ago, that was a big improvement, but not very detailed. So in the late 90s, some folks up in Oregon, and I was up in Oregon at the time, started using a submersible to understand fish populations off Hecata Bank in Oregon. And the first study, the first year, was headed by Bill Piercy, and the next 3 years were Dave Stein, Mark Hickson, and Brian Tissot, which many of you who've probably heard of in the past. So the interesting thing, and this I think was Brian Tissot's concept, was they tried to identify the fish that we saw with a very small scale habitat or bottom types in terms of the type of rock or sand or mud that we were going over as we were looking at the fish out of an old submersible called the Mermaid 2, which isn't around anymore, fortunately. And then later in that decade, Moss Landing folks, all of these people were associated with Moss Landing, headed up by the geologist Gary Green, got together on a ship, or on ships, because it took a while to complete, to come up with a global classification scheme for substrates relative to fish. So we would call them habitats for fish. And we tried to scale it from the mega habitats down to the micro habitats, so that we could partition what we saw into an appropriate level independent of the vehicle that we were using to observe the fish. And we came up with macro habitats and micro habitats and some modifiers, and even threw in some of Amandicons and vertebrates to make her happy. And then at the end of that decade, we started seeing a boom in underwater vehicles, visual vehicles for identifying fish and habitats. And the sub that I used for a couple hundred times was the Delta sub. Used the one-person deep rover for a while. We have lots of ROV footage, and then we developed here the Boss video lander, the BRUVs, and then Mambar uses AUVs. So the last 20 years, we have extensive video information using those tools. So the first thing that I wanted to do is to say, what are we learning from these submersible surveys relative to fish and their habitats, and do landscape ecology principles apply? Thinking that if they were the same as are occurring in watersheds on the land, that we'd be ahead of the game by applying those same tools. So Moss Landing student Kristen Hunter-Thompson, who's now up at UC Santa Cruz, where their husband Malin Pinsky, Malin graduated from Stanford, went to Princeton, I think, and then now is back at UC Santa Cruz. And Kristen tried to use some of our submersible surveys to see if she could identify the same landscape principles. So the first one we looked at was the proximity to edge. So the idea in the land is that you have animals on the edge at the habitat ecotone that are going to show an increase in density, species diversity, and size of individuals, because they can rush out and feed in a variety of different habitats and take shelter in the appropriate shelter habitats. Also, because of that, the patch shape that is corrugated is going to have a higher density, diversity, and size of individuals than the same area, but with different ruggedness of its shape, for the same reason that animals can take shelter here, then dash out and feed. And then similarly, the patch size paradigm is the higher area of habitat is going to see an increase in abundant species richness in the size of individuals. So Kristen used some of our submersible surveys off of Point Lobos, and she identified different sizes and shapes of habitat patches to see if what we saw from the submersible fit the landscape paradigms. Well, answer was no. As you might have guessed, we saw no difference in the density of biomass or the assemblage between the edge and interior zones of the rocky bank patches, but there was some increase in the deep rocky areas. So that already is giving you some idea of the differences, maybe akin to elevation along a mountain slope, that you're going to see different patterns, not necessarily different animals, but different patterns of the combination of animals along those clines. Patch shape was not a predictive indicator, and the size of the habitat patches was a greater factor in explaining assemblage structure than the shape of the habitat patch. And we were starting to scramble and ask our head, well, why don't those paradigms work in the ocean? Well, the answer is pretty simple. It's the water column. Unfortunately, that's not an easy thing to deal with. But that's, I think, is the answer. is that we need to figure out, and this, I'm jumping ahead to the end of the talk, we need to figure out some new paradigms to integrate the water column with the benthic habitats. And I think we're going to need new tools and new approaches to measuring those differences. I would love to put a camera out on some of those rocky patches and let it sit there for a year and see how the assemblages change through time, and of course with some current meters to see where they're going, what's happening there. Moving along, Mary Gleason of Nature Conservancy and I worked with Mabari. Chad Kesey was the lead engineer, and Ryan Fields was a student here, if any of you remember him. We worked on developing the BOSS tool with the idea of being able to rapidly move it dozens of kilometers apart from each other so that we could do point sampling rather than strip samplings with submersibles or ROVs, just to give us a different kind of coverage spatially. We use these tools, again, to look at the bottom habitats and look at our species and how that relates to the bottom topography. And we found that there were some very clear similarities among different sites. And you can see where these are different general areas along the coast. And there were specific areas that were close to or similar to one another in their communities. And they all had different kinds of bottom types. And you can see some of the coverages here in terms of the bottom type and the species down this direction. But just focusing on the colors, you can see that there are areas that are similar, and then there are areas maybe adjacent that are very different in terms of fish communities. And those differences are due to depth, latitude, currents, et cetera, et cetera. But what we're trying to get to is, again, from the terrestrial perspective, can we predict what we're going to see when we go there? Well, we can predict the species pretty well, but we're not necessarily able to predict the exact communities or the sizes of the fish. So, and the reason is that this is from BOSS data, and also Milton Love and Mary Yaklovich did a lot of submersible surveys over the years. There are with differences among hard, mixed hard, and soft that are just pretty variable depending upon the species. And the reason is that you'll see some animals like canaries, rockfish, who I think of live on high relief rock, and yet they show up in high proportions on sand bottoms. And we know now from some work that Heather Bolton from CSUMB did that is because those are juvenile canaries. And Rick Kvitek from Moss Landing, CSUMB, wrote a paper in the late 90s, I think, early 2000s, showing that juvenile canaries were living on ripple scour deposits on little sand waves, nowhere close to where they live as adults, both in space and time. And so these differences, as the animals mature, there are changes and ontogenic shifts into deeper or different types of habitats messes up the statistics. Damn statistics, right? So we need to get finer scale information so that we understand those differences in habitat use at different ages. And I'll come back to that in a second. The other thing that we're looking at is the habitat complexity. Is that a big lump of rock has some relief that makes it different than a smaller lump of rock, but the voids in the rock, the holes where animals can hide, and the rugosity, or how crenulated the rock is, make a great difference in what you're going to see. And so Ivana and I have had a number of different conversations about this, and one of my thoughts, which I haven't pursued, but is does the fractal dimension of the rock matter? In other words, if you have one big smooth versus lots of little craggy rocks, are you going to see different things? And that's an area to pursue, I believe. However, just about the time that we were starting to figure out these observations of fish and their habitats, multi-beam came along. And we thought, well, gosh, we're going to have all we need from the multi-beam, because here's a computer mapping program that can give us a variety of different attributes to measure instead of is it rock, is it sand, is it big rock, is it little rock. Here are all kinds of different topographic variables that we could get from the side scan sonar or from the multi-beam sonar. And we've gotten really good information from that. So we have here are maps of soft substrate versus hard substrate. And you think, well, that should give us a really good information. And in fact, there have been people who have suggested that we use that level of map to estimate population abundance of fish along the entire coast. Because we've got most of the coast mapped, we should be able to get a biomass estimate. We also have that multi-beam to look at a seascape scale, just in case that's a better scale than the highly detailed bottom type. And we can develop, based upon the classification system that Gary Green led, we can look at mega and meso habitats as well as the micro habitats. So we have some information that's been developed to estimate habitat suitability or likeliness of occurrence of fish or other marine organisms. The map on the left here is predicting the probability of occurrence of Boccaccio along the coast based upon our estimates of the likelihood of seeing Boccaccio in places. And then that's occurrence. And then we also have catch per hour or some estimate of relative abundance using these maps. And the idea is that we can use these maps to predict fish distributions outside of an area that was sampled, identify areas for a habitat. I find myself looking at that bug and thinking it's the laser. You know, what? Um, and we can obtain species density and biomass estimates, use this. So Mary Young from UC Santa Cruz tried to do this at Cordell Bank. And we have a number of different submersible surveys. You can see these black lines are where submersible surveys come from. So she used the multi-beam maps and species distribution methods to map out the probability of occurrence of, in this case, yellowtail rockfish at Cordell Bank. And Heather Bolton's thesis, it's from CSUB in 2014, did the same thing with juvenile and adult canary rockfish up here off of Point Reyes. And also, Megan Bassett, for her thesis, and James Lindholm were able to identify a species distribution, a species probability of age 1, age 2, and age 3 plus lingcod, because we know that lingcod settle out in sandy areas and move into rocks. If they young settled into the rocks, the other lingcod would eat them. So we see this ontogenic movement from sandy areas here in Monterey Bay into the rocky areas and deeper. So that was a pretty good approach to try to identify the abundance of those things. And then Lisa Wedding and Mary Yakovlevich of NIMS took the same approach to look at the biomass for rosy rockfish around Point Lobos and Point Sur. So that was exciting to me. And I thought, well, gosh, we're zeroing in on the answer. So Moss Landing student Ann Tagini wrote her thesis on comparing the species distribution maps at Portuguese Ledge. Everybody know where that is off of Point Pinos. It's for those of you who are interested in deep water, this is an MPA, really, really cool area with rocky habitats surrounded by sand that has a ton of fish and was a long-time fishing area, but it's a really good study area because it's so close. Any rate, Anne wanted to ask, based upon the mapping, and our surveys using the BOSS and the submersible surveys we've done here, what's the comparison? How well does the multi-beam and species distribution maps match what we saw when we're underwater? And the answer is not well. The predicted densities were always greater than the observed densities, and the models underpredicted densities where we saw fish and overestimated densities where we didn't see fish. And you can see this from this graph. That's the one-to-one correspondence, and the predicted densities are all down here in the higher density than what we saw. So that caused a problem. And then another thing that caused a problem was the scale. The scale at which you chose to map influenced the results on your biomass. So if she mapped in Portuguese ledge at a 1 meter level of resolution, then the biomass of copper rockfish was estimated to be 1,300 tons. If she did the same model, but used it at the 5 meter level of resolution, the biomass was estimated to be 75 tons. And at the 10 meter level, the biomass was estimated to be 25 tons. So that's a order of magnitude difference just based on which scale you take. So that made me scratch my head and say, well, what's right? What's true here? And I don't know the answer. But that's an area that I think needs to be evaluated in the future, is what's the effective scale. I don't know if I have a slide on it, but Kinsey Matthews, who just graduated last year, and hopefully some of you saw her thesis defense, she ran into this same problem when she was modeling species abundances along the coast. The scale at which you used for the mapping, for the area delineation, greatly influenced the abundance estimates. So I don't know which is right, but that's an area for investigation into the future. Here's the other thing that Kinsey found out, and this will be probably too complicated to go over, but Kinsey developed a model for each of these years of information, and she found out that if you change your test year, because everything in this is testing against a predicted estimate, the year you choose for your predicted estimate influences the results of the model. So now we have a scale of the map influencing your outcome, and also the time of when you choose to use as your basis, as your baseline, influences the results of the model as well. So that's another area that's ripe for investigation. And we have information, so anyone of you might be inclined to do some modeling mathematically, the data are there. The other thing that we learned, and Kinsey wrote a paper about, is that the location, all of these are different locations ranging from San Francisco all the way down to the southern Channel Islands. And there was quite a bit of variation in estimates of abundance by site. And so, these are 3, 6, 8 different sites, but this is probably a 400 or 500 mile stretch. I wonder if we were to pick 8 different sites in that 500 mile stretch, if this is what we would see, or if we would see again something that's totally different. So there's a lot left to go before we can say that we can reliably predict what we're going to see based upon our current models. Now, there's some hope here. James Lindholm, Carrie Bretz have been working with the statewide mapping project to look just at the rugosity of different areas. And they do think that they've found some relationship between a relative abundance of a number of different species and the high rugosity rock habitat. So we have a proposal in right now to see if we can articulate that better to see if it's true or not. And then Jake Todd just gave his thesis defense a few months ago, and I'm not going to spend a lot of time on that, but just to say that, again, these are, this blue area are the shale beds off of Monterey, and this green and the red areas are different areas in Carmel Bay. Carmel Bay is a higher relief area, different rock type, more granite rock than sedimentary rock. And Jake found a difference in the species using the NMDS plots, and that depth topographic positioning index, or local relief, and vector ruggedness measure, the crenulations of the rock, all made a difference in what we saw there. And those are relatively subtle differences in the rock habitat. And so another question is, how sensitive are our models going to be to what appears to be important variables? If you just translate this concept from the mapped variables into species, you see there are also species differences here. So we still have a long way to go to pin down what species are living in and what rock type when we start considering these finer and finer level of defining what a rock is. And Jackie Mohe also gave her thesis defense in December. And what I wanted to show you here was that the measure of the essential tendencies give us a pretty interesting look at the diversity of species from hard to mixed hard down into soft. And so that's really, really good news. But look at the variation, around the mean. So that's pretty large variation around the mean. And if we're just talking about central tendencies, I think that we're pretty confident in this. But then when you start looking at any given species, that might change that perspective. And the same thing was true in Katie Sierri's thesis. She did some work we were doing in Spain. But notice the high variability in her samples relative to these benthic categories we were coming up with. And of course, one of the reasons, this is Jen Chu's, from Jen Chu's thesis, is that if you look at just 2013 versus 14, these are the animals in the diet of yellowtail rockfish, and huge difference from year to year. So of course that was due to the heat wave that we had back then. And so, just another variable to toss in the mixture is going to be the water column dynamics, as we're trying to identify a species habitat association. Habitat is important, but also is connectivity. And understanding the connectivity among habitats is really important, but difficult. You have first of all, the random pattern of settlement of juveniles, and that is going to affect what we see, where we see them as adults. You have ontogenetic movements, and there are seasonal feeding and reproductive movements. There's diversity of species and life history strategies. And then there's a famous ichthyologist who once told me, fish do whatever they want to. So the ichthyologists from Moss Landing and Greg Caillet studiously avoided movement studies because they weren't as precise as his stomach content studies. But if we start looking at movement studies, we've learned a lot over time. Whoops. This is some pictures from a study I did in Alaska with Tori O'Connell of the Alaska Department of Fish and Game, where we tagged 90 lingcod on a pinnacle off of Sitka. And this is a picture she took from a submersible showing how they're just laying like cordwood up there. But the tag data showed the mean depth by month for male and female, and you see that they flip-flop in terms of what depths they're at, except that in the fall when they're gathering up to mate. And so when you observe is going to affect your expectation of species habitat associations for migratory fishes and for different sexes. So we need to account for that. Similarly, Kristen Green and Ashley Greenlee, who finished up their theses here in about 2014, we had a big project. We were tagging fish in Carmel Bay, and we tracked their movements for a 2.5-year period. So these are 31 acoustic receivers that we put out in Carmel Bay, and their expected zones of reception. And we tagged black rockfish, blue rockfish, lingcod, cabezon, and one other species that I recall, and measured the activity space. So this is the blue rockfish, and as most of the literature talks about, they have relatively small home ranges. So that's going to be really good for blue rockfish to predict a habitat association. And as most of you divers know, kelp forests are probably the best indicator for the habitat association of blues. For lingcod, they were occupying mostly high relief habitats. And for black rockfish, they were occupying a mixture of high and low relief habitats. However, we found that the black rockfish would move out offshore during the day and move back inshore in the evening. So they're living and working in deeper waters during the day than we find them at night. The vertical movements were significantly correlated with wind speed, atmospheric pressure, the square of the wave height, and the upwelling index. basically when it would get rough inshore, the fish moved deeper and offshore. So relative to species habitat associations, now we need to describe both of those habitats. Where are they going to reside in calm days? Where are they going to reside in rough days on the ocean? Also, we found from those tags that rockfish make a northward migration. From our study area here, we had one tagged rockfish go up to Nia Bay. And all fish were last detected during major storms. So what happened was a big storm would come, and this is my interpretation, they'd jet offshore and then get carried north by the prevailing winter currents as they were advected north. And we can see that. Well, let me go back. This is the track, the CCFRP tag returns for black rockfish. Again, showing movement all north. And what we've learned from our colleagues at NIMS is they do plankton tows, juvenile plankton tows, and they see an aggregation of juvenile black rockfish down here in the southern Oregon, northern California area. So what we think is happening at these juvenile black rockfish are migrating up here as they grow and spawning up here, and their larvae are being advected back south. So again, that there's more there from a habitat perspective to describe. Cindy Dawson worked on her thesis on prickly sharks. It's a relatively rare deep-water shark that lives in the submarine canyon offshore about 3 miles, sits on the bottom during the day and does squat. As far as we can tell, they didn't move. At dusk, like clockwork, they would start moving up from 200 meters deep into 5 meters deep, right here off the mouth of, near the outfall of the Moss Landing. And that's how we first learned about it. It was people diving around the outfall and seeing these sharks. And then they'd be spending the night up here, moving all over the place, very active, feeding, and then head back down canyon during the day. So the time of day is important there, but that's where all the feeding was being done. Similarly, Aaron Carlisle, who's now a professor at Princeton, he was here last fall, I think, to give a seminar, tagged leopard sharks. And leopard sharks were very active at nighttime and stayed up in the slough areas in the backwaters during the day. Another project of mine down in Belize, where we were working to tag Nassau groupers, Enrique Sala and Kiki Ballesteros from Spain, got me involved in this, and we caught groupers up here who were spawning, and then we, with the long-term acoustic tags, and released them and caught them traveling back up here a year later to spawn again. So the interesting thing about this, and something we don't know about with our species here, was the co-occurrence of timing of things. They were on these NASA grouper were on the spawning ground, and this is the hour of the day for 2 different days a year apart when they were spawning. They were at shallow water, shallow water, shallow water until just about noon, and they dove deep to 100 plus meters. And that was the first evidence of NASA groupers being down that deep. If you read books now, you'll just see them, they lived to 60 or 80 feet. But they were, when they got finished mating, spawning, they dove deep and they stayed there. And, oops, pressed too hard. And then they stayed deep for about 2 months, and then the same hour, the same day, came up to the surface. And then this is the next year, same thing. They all went deep after spawning, stayed deep, and then came up to the surface on the same day. So there was synchrony with the spawning evidence, spawning patterns or behavior. And, I'd like to know if that we see that same type of thing in rockfishes. Because as a group, often we see rockfishes go down at night into deeper waters. So those are all the things I've worked on. But we have a lot more information to gather relative to large animals and we know the white sharks. This is a tiger shark picture I took off Cocos Island a couple of years ago. But we know we have all these juvenile white sharks up in the water column near the beach up in Capitola area. And Dave Ebert suggests that they're there sunning themselves to gain body temperature before they go back down deep to hunt. And, it'd be interesting to find out if that's his hypothesis is true. I don't have any reason to suspect it isn't true, but it'd be nice to verify that. So that's the progress that we've done around here with mostly students from Scott's lab and my group to learn about species habitat associations. And, we know that we need to do more and correlate that those movements with water mass movements, water changes, habitat changes, and then again we're behind the terrestrial folks because who knows what our climate's going to be, our environment's going to be 20 years from now, and how will that affect the distribution patterns, the movements of the animals that we have off our coast. So there's a lot left to learn and put it into a conceptual model of occurrence and movements relative to the changing ocean. And that's it.
Okay, we can take questions. Your voice will be heard over the interwebs, but not amplified here. So just raise your hand and I'll come around to you.
Oh, I thought I was getting a get off.
Non-marine scientist with a question.
Yeah.
So you showed those surveys where you measured the biomass at different spatial scales. And I'm trying to understand why, when you looked at it at 1 meter scale, you saw 1,000, was it 1,000 kilograms of biomass, and when you move to the larger scales, you don't. Why do you think it is that you're missing all that biomass when you go to bigger scales? What is it about that measurement? I'm perplexed.
I am too. Nate might have a better idea than I do.
I think, in a simple sense, it comes down to like a mathematical artifact. It's like inflating the uncertainty as you step into more coarse grain resolution.
So you're just missing fish as you go to a bigger scale.
So there's a trade-off, because if you go too precise, then you're overestimating.
Right.
Well,
In all likelihood,
yeah.
That's how you calculate roughness. Yeah, so it's how you calculate roughness. And so using different pixel sizes, you come up with different numbers. So as Rick was saying, roughness is not it's spatially correlated. So something can be very rough, like this carpet here, at a millimeter scale, centimeter scale is very rough, but at the scale it's flat, right? So that's, yeah.
So you're using surface area as a proxy for biomass?
You use, well, one of the correlations between its roughness, which is a ratio between surface area and planar area. Yes, indeed.
When you get to 5 meter scale, you don't see that.
Right. So the goal is to come up with the scale-independent roughness method, which is fractal analysis, which is multi-scale approach. Thank you, everyone.
I have another question about that. So what were the data that were fed into the model taken at? What scale? That you had to start with some original data. So what were those, and were those taken at multiple scales? Or maybe
No, that's a good point. And again, that's something that Ivana would be better suited to address. But we're taking it at one scale, which is the scale of our video unit, be it a BOS or a transect off of an AUV or ROV. So, and that scale is on the order of 2 or 3 meters by a meter and a half or 2 meters. And the roughness scale is from the sub-millimeter level of the echo sounder, the multi-beam, that's just measuring every, whatever it is, centimeter of ocean floor, and then aggregating up to whatever scale you want it to be. So it is a mismatch.
I have a brief question.
Okay.
I was curious about the strong temporal variability you noted, and how it matters when you sample the fish population for making predictions. And so you showed a lot of variability depending on which year you pick. That was part of a student thesis, and I was curious what were some of the kind of leading hypotheses for the process that was driving those interannual trends.
So this gets into something I didn't talk about, but and I don't know if I'm way off base or not, but I don't think I am. You've got a rocky area that surrounds this room, and when you go out and measure June 14th this year, we've got a strong northwesterly wind with a current advecting across the rock area here. Maybe a lot of the fish are on the lee, hiding out of the current. The next year, you're a month earlier, and the current's going the other way. Now the fish are in a different spot. Or there are fish, there's a lot of zooplankton being advected across the rock from the north, and they're on the leading edge. And so if you have a transect-based method, you're integrating across all of that area with lots of zeros and one area that has a bunch of numbers. Well, then that gets into how well are you sampling this rocky patch in here. And that's why I think that I'd like to see a camera put down on a rocky patch, or cameras, so you can see where the distribution, how the distribution of fish changes over time. Because I think that's where a lot of the variability comes from, is what month are you sampling, what hour are you sampling, and what are the current conditions.
Any other questions? All right, and please join me in thanking Dr. Starr.
Thank you. Thank you. Yeah. A lot of interesting. I wish I had another 30 years.
I know, right? Yeah.
Because
There's not enough hours in the day.
Yeah.
So many cool patterns.